Hello, students. Welcome back. Here we are on a lab day, but before we get to any activity, I just wanted to mention a couple things here. I'd like to finish up our notes real quick from page 58 to reinforce the importance of meiosis. So turn to that page if you would right now, page 58 in your book, and let me tell you a little story. In fact, it's a love story. A love story of Goober. So let's look at Goober for a moment. And Goober is a soft, cuddly little critter. Perhaps you've heard of goobers before. Well, this is what a goober looks like. And a goober has four pairs of chromosomes. One pair of chromosomes, the red ones, determine the fact that he's got four legs. The green chromosomes determine that he's got two eyes. The blue chromosomes determine that he's got one tail, and the black chromosomes determine the type of fuzzy fur that Goober has. Now, as you can imagine, Goober looks a little lonely here. So, as Goober is wandering through the field, one day Goober sees his life partner, Goobette. And yes, it is love at first sight. Goober and Goobette were made for one another. Ooh la la, they love each other, they get married, and they produce children. So they make lots of little goobies. So Goober and Goobette get together and make goobies. Now, here's the deal. If things are going well, in other words, if meiosis has occurred correctly, all of those little goobies of Goober and Goobette will look lovely and normal, and they will look just like Papa Goober or Mama Goobette. However, things don't always happen correctly. So let me ask you some questions. What do you think would happen if all the chromosomes from both parents were combined? Or what would happen if some of the chromosomes were missing in the offspring? Do you have any idea what would happen with those goobies? Well, our little goobie might inherit some mutations, because if they don't have the right number or the correct number or the right alignment of chromosomes, well, funky things can happen. And a goobie's morphology or its physical appearance will be unusual to say the least. In fact, it might not even survive. So, my question to you is, why is meiosis important? Hopefully, you gave some good logical answers as to why it is important, basically to prevent mutations. Now, some mutations might be good, some mutations might be interesting, and some mutations might be harmful or fatal. Check out some of these real life mutations of various animals, like a two headed turtle or a two headed snake, or multiple legs on goats or frogs. You know, some of these mutations might make interesting anomalies in the animal kingdom. Oftentimes, some of these mutated individuals will not be able to survive until adulthood. But as interesting as these mutations are, sometimes mutations do happen among human beings as well. And sometimes these mutations are the result of chemicals in the environment that alter the natural flow of meiosis. And strange and unusual things can happen to those chromosomes. And so they can happen in nature, natural mutations, but they can also be human produced. So, why is meiosis important? To prevent the production of mutations that might be damaging or deadly to an organism. Now, with that in mind, I'd like you to turn to the classroom activity on page 54. And the first thing I'd like you to do here today is to complete the classroom activity on page 54. It probably will not take you all that long to complete it, especially if your notes from yesterday were really good. So, completing the classroom activity, I want you to work through it on your own. Read the problem and write a hypothesis before you begin it, and then follow the instructions as you complete the classroom activity. Now, if you have remaining time this period after working on the classroom activity, I have provided you your own individual scissors so that you do not have to share scissors. And what I'd like you to do for the remainder of your time after completing the classroom activity is actually remove page 55 from your book. You can carefully rip it out of your book if you want, or you can use your scissors, but you're going to remove page 55 from your book. And it is a little puzzle for you, a three-dimensional puzzle for you to create, which is a model of DNA. Now, the instructions are found on the back of the template for your model. In other words, they're found on page 56. So I would encourage you to read page 56 before you start folding up your model of DNA. But when you get to the end of the activity, you should be able to construct your DNA model, your paper model, and it should look like this. Now, if you should not have time enough to complete your model during the period, I encourage you to bring it home. Try to finish it up at home and bring it back tomorrow. And maybe if we have enough really good strands of DNA tomorrow, maybe I will sanitize them and we can tape them together to make nice long strands of DNA. Otherwise, you can just keep it yourself and you can hang up your own little model of the twisted ladder, the double helix of DNA, and check out how the nitrogen bases pair up. So today, you're going to, again, finish up the classroom activity first, and then for the remainder of your time, work on your model of DNA and have fun completing it at home this afternoon if you don't finish it in class. It is a bit of a challenge, so take your time and follow the instructions, and I'm sure you'll do fine. But until then, I will say bye-bye.